Hello everybody. Today we'll start the first lecture of my course. The course title is Essential of Biomolecules, Nucleic Acids, Peptides and Carbohydrates. As I have talked in the introductory video, that in this course we'll cover the importance of the biomolecules, how the molecules present in our body, they are involved in many biological processes, how an enormous amount of chemistry is involved in those biological behind those biological processes, and what are their structures and functions, and how could we modify later to alter their activities. So, in this course I will be covering two textbooks, or some of the chapters should be picked up from these two textbooks. One is these references. First one is the Essentials of Chemical Biology, which is by Andrew Miller and J. Tanner, and then second is of course the Biochemistry, which is written by Berg, Zakow and Stryer. Before starting the lecture, I would this is how our campus looks like. I belong to IIT Guwahati, and this is what the campus looks like in flowering season, and it's surrounded by many hills and we have few lakes inside, and this is the structure of it. I belong to chemistry department, and for those who are interested, I would just tell you what are the programs we run from our department. Now, one is of course the 4-year BTech degree, that is in chemical science and technology, and if you want to come to that, you have to appear for IIT JEE Advanced. Next is we have 2 years masters program, MSc in chemistry through JAM. And we have of course the PhD program, which we recruit from candidates who are eligible with GATE, CSIR and or INSPIRE fellowship. Okay. So, why this course? So, this course is interdisciplinary between the organic chemistry and biological chemistry. So, organic molecules, why they are important in biology? First of all, of course, is to understand the biological processes. What are the processes that are going on in our body? That is a curiosity, perspective of curiosity, that if we want to know, and of course it is a long curiosity that exactly what is going on in our body or in other living cells. So, understanding the biological processes. So, in order to know that, what you have to know, we have to understand what are the chemical reactions that are going on inside the cells, what are the molecules involved, what are the enzymes involved, and how they react for that specific kind of reactions. And you can see that many of those reactions that happen in biological cells are almost impossible to produce in laboratory. So, they are very specific reactions, or they are very specific organic transformations that happen in living cells in presence of other surrounding biomolecules such as proteins. So, once you know what are the reactions going on, what are the processes going on inside the living cell, then you can find out the changes of those reactions that happens in diseased cells. So, you can understand a disease, why a disease is formed, and then you can diagnose a disease using other chemical tools or organic chemistry tools. In modern days research, we have our diagnosis techniques have been elevated to a good level, and we have lots of kits now available, many different types to diagnose a disease, and most of those kits actually came from the chemical reactions. So, understanding the biological process, and then you can find out about the disease. Now, once we know about the disease, then we can find out what are the ways to rectify that. What are the ways that will rectify the anomaly in the biological reactions back to the normal way. So, that is where the drug discovery comes. And of course, most of the drugs are the molecules that are developed in the laboratory or isolated from natural sources, and many of them are organic molecules. So, in order to develop a drug compounds, of course, before that we have to understand all of the biological process, and we have to understand what happens or what is the anomaly that happens in the diseased cells. Then we can find out how to stop that anomaly. So, that is a drug discovery thing. Now, once drugs are there, once you find out a drug is active against certain disease, that is not the end of it. So, the next challenge is to take the drug inside the diseased cells. That is what we call drug delivery. So, what happens in our body, our cells are very well protected. They do not allow the foreign particles or the foreign entities to cross the cell membranes or to get into our body. That is why the cells are very well protected. Otherwise, every day we are exposed to so many stuffs, but most of them do not penetrate our body. That is how we are all still alive. So, drug have to be taken into the cells, and that is what is called drug delivery. And here comes the in modern days, large part of the drug delivery comes from the nanoparticles or nanoscience. We can develop lot of different kinds of nanomaterials where you can embed the drug inside or encapsulate the drug inside it, and then it has higher probability to get inside the cells, because our cells, although I say that are very well protected, but the cells have spaces in between them. They are called the pores, and they are very small pores actually. When you take the nanoparticles, they have a very smaller size. Typically for the drug delivery, if you use nanoparticles which is between 100 to 200 nanometer range, then they are small enough to cross through those pores. So, it is basically fooling the cells. Cells will not understand that something is getting into it. So, that is how you can take the drugs into the cells, the foreign particles into the cells. And the last one, but not the least, nowadays we are aware of the polymers and plastic materials. We have most of our materials that we use every day and surrounding us are made of plastics or polymers. So, now we are changing that to a biodegradable polymer. So, you have to find out ways how can you make the polymers, or how can you make the bonds which can be clipped naturally over time or by using enzymes that will degrade the those molecules. So, that is the biodegradable polymers. So, I wanted to give you the brief idea of why this field of organic chemistry and biology is important to understand. So, these are the ways you can play with. All right. So, this is a molecule. This is an organic molecule actually. It is a large molecule. Can you recognize this molecule? Can you recognize this organic compound? Well, this is paclitaxel or taxol, a very celebrated organic compound. This is one of the most effective anti-cancer agent that is there in the market. So, if you look at the molecule, this molecule is very complicated and it has around 11 chiral centers. You see, 1, 2, 3, 4, 5, 6, this is not chiral, 7, 8, 9, 10 and 11. So, it has 11 chiral centers, extremely hard to find out even the structure of it. So, this is paclitaxel, which was originally isolated from a tree which is this Pacific yew. It is a very large tree, very old tree, usually 100 years old, and this molecule taxol is obtained, paclitaxel is obtained from the bark of this tree. It was originally isolated back in 1969, where if you use around 1200 kilogram of the bark, then the crude amount of extract that you can isolate is around 28 kg. And from that, after processing, you get only 10 gram of pure material, pure paclitaxel, which is the anti-tumor drug. Now, so you can understand the tedious of this process. From a whole 1200 kilogram of the bark, you can get down, after lots of steps of chemical processing, you can get down to only 10 gram of the pure material. And the dose, typical dose, depending upon the type of cancer, typical dose of paclitaxel is around, maybe it varies a lot of course, but 150 to 170 milligram per dose, and that runs for weeks and weeks. So, 10 gram of pure material is enough for only few doses, and obviously the necessity is to synthesize this molecule in the laboratory. So, that is why the natural products are coming as drugs, and then of course, the source of the nature is not good enough to meet the needs of the patients. Then the necessity comes that we have to synthesize these compounds in the laboratory. And that is a very tedious job, of course. So, for this particular molecule, after a lot of research, hard work, in 1994, Professor Casey Nicolau and Professor Robert Halton, they independently found out or reported the total synthesis of this whole molecule Taxol. So, this is plant alkaloid, which has of course 11 chiral centers, and this is used in many types of different types of cancers, including breast cancer, cervical cancer, ovarian cancer, and so on. So, for the medicinal chemist or for the natural product chemist, the process is something like this. Most of the drugs that are available in the market, many of them are actually originated from the natural sources. And how they came into the drugs? So, what people do, what scientists do is they first isolate the compounds from different trees or the plants. Like the alkaloids, terpenoids, and heterocycles, other molecules. So, they isolate the molecules from the natural source, and then they go for testing, biological testing, to see if these molecules or some of these molecules show any positive activity towards any disease cells. So, once you find out that a molecule that you have isolated is showing some response to a disease, then you go for more isolation and then elucidation of the full structures. That is really a tough job and very tedious process. And after that, then it comes to the, after different stages of verification, it comes to the market. So, lot of organic chemistry is involved in finding out about a solution of a disease. Next is the understanding a disease and design of a drug. So, if you look at the tumor or the cancers, tumor is basically the development, or cancer means the uncontrolled divisions of cells. If you have one cancer cell, then very quickly it grows fast and it makes more cells, and they are all infected tumor cells. And almost all cancers, I will see later on how they are formed. Almost all cancers are due to mutations of DNA which comes from the damage of the DNAs. So, this is a DNA. So, every day we are exposed to a lot of external and internal agents. So, therefore, our DNA also are exposed to a lot of internal agents as well as external chemical agents. Internal chemical agents means it goes through metabolic processes, and during metabolism a lot of radicals are produced, like peroxide radicals and all. So, they react to the DNA nucleobases, or we are exposed to chemicals, we are exposed to radiations, for example UV radiations or pollutants. Many of them actually what they do, they change the structure of the DNA. I have just pointed here. They change the structure of the DNA, and in turn their base pairing gets changed. So, once these structures are changed, now we have a very good mechanism system which actually repairs those damages. These are called DNA lesions or DNA damages. DNA lesions or damages. So, the statistics is around 10 to the power 6 number of lesions are formed in each cell per day. So, around 1 million of DNA lesions are formed in every cell every day. You can imagine how much amount of damages that happens inside our genome or in our DNA. But we have a very good mechanism to repair those lesions back to the normal. So, usually all the lesions are repaired back to the normal DNA. But if they are not repaired properly, then there are the consequences, two consequences. One is it can either stop the DNA replication process, which means the cell death, or you can undergo mutations, or it can induce mutation in the genome, which means basically is the reason for aging or cancers. So, once we know the chemistry behind it, then only we can understand what are the mutations happen or how to alter those, how to design a drug once you understand a disease. So, here I will show what is the chemical process that goes on in our normal cell as well as tumor cells. As I have said that DNA is the key material that is required for cell divisions. So, how is DNA synthesized in cell? One of the ways is it starts with uracil. We have uracil in our body, uracil or uridine with a deoxyribose sugar. So, uracil is transformed into thymine. So, in uracil you can see here there is a hydrogen H atom, and in the thymine this H is replaced by a methyl group. So, this is the only addition and this is the chemical reactions. A CH bond is replaced by a CCH3 bond, and this reaction is catalyzed by the enzyme thymidylate synthase in our body. Once the thymine is synthesized, it is involved in synthesis of DNA, and then DNA undergoes cell divisions. Every cell has DNA, so DNA multiplies when the cell multiplies. So, that is how the cell division happens. Now, in tumor cell also same thing happens, only thing is that it happens very rapidly, faster, much more faster than the normal cells. So, one way to block the cell divisions or to block the number of tumor cells is that if you can block this reaction, this transformation uracil to thymine, which is catalyzed by thymidylate synthase, and here come the chemistry. So, thymidylate synthase requires a substrate which is uracil. It is very specific, it can take only uracil. Now, this molecule here, if you see 5-fluorouracil has a fluoride atom here, where there was H hydrogen atom at the uracil. In 5-fluorouracil you have a fluoride atom. Now, hydrogen and fluoride atoms both have almost same size. So, overall they do not change the size of the molecule, and therefore, it is expected that 5-fluorouracil also can dock inside the active site of the thymidylate synthase. In other words, 5-fluorouracil can be used as a substrate for this enzyme. Now, once the 5-fluorouracil is taken or recognized by the thymidylate synthase, then the difference is the fluoride atom. Now, the CF bond is very difficult to break, and of course, the enzyme does not know how to break a CF bond. In this transformation, enzyme knew how to break the CH bond and make the CH3 bond, CCH3 bond. So, once the enzyme takes up 5-fluorouracil into its active pocket, then it cannot do the further transformations, because it cannot break the CF bond, and therefore, the thymine cannot be synthesized. So, synthesis of thymine is blocked. Therefore, the DNA synthesis will be blocked, and therefore, no cell divisions, and tumor cells will be slowly dying. So, that is how this molecule was developed, that you can develop a new molecule which will interfere into the biological reaction. We call them inhibitors. So, that is one way. A second way is uracil to thymine transformation happens fine. We do not do anything here. The next one is the synthesis of DNA from the thymine to the DNA synthesis. Now, once this DNA is synthesized, this is the structure of the DNA. This is double helical structure of the DNA. I will explain later in detail. Now, if we can block or if we can inactivate the DNA itself, then it cannot do the cell division. Such is a molecule called cisplatin. This is an inorganic complex which is also a renowned anti-cancer agent. 5-fluorouracil is also a commercially available chemotherapeutic agent, so is cisplatin. So, cisplatin what it does, it binds at the core of DNA. I will show later how it exactly binds. So, it binds, it forms a metal complex within the DNA, and therefore, that is a strong binding, and therefore, it cannot undergo further replication process. It is basically blocked or it is dead. So, DNA is becomes dead and it cannot produce more cells, therefore, tumor cells will be killed. So, once you know the reactions, once you know the chemical ways, once you know the number of enzymes or what are the enzymes involved, the structure of those enzymes or other molecules involved in a reaction, then only you can figure out what are the changes you can make or what are the new molecules you can produce to alter those reactions. I will show you and give you another example of how understanding a disease is important. So, another very notorious disease is, of course, is Alzheimer's disease. It is a progressive neurodegenerative disorder, damages the brain cells or connections between the brain cells. What is the cause of the disease? What is the reason? The full reason is not yet known even today. So, one of the reason is the aggregation of the protein or the particular type of protein, amyloid peptides, we call them peptide, beta amyloid peptides outside the nerve cells. Because it is a beta structure, they are actually the flat structures, so amyloid proteins, and they get stuck onto each other, forms an aggregation, and once this aggregation are formed, they cannot move, they cannot do the biological functions. It is like a stone formations. So, this is how the process looks like. This is the native A beta peptide structures, open form. Now, if it is misfold and then forms a oligomer, many of them come close together, forms a oligomer, and this is the fibril formations. Because of these oligomers will be stacked on top of each other and forms a fiber basically. Here you can see in this image and the fibers are formed. So, the fibrils are formed, and then that is the reason for one of the key reasons for Alzheimer disease. Now, once you know this process, you can find out ways how to disrupt those. If we want to stop this aggregation, if we want to stop the fibril formations, for example, this is an example of you use a molecule which gets into the folding, and then it does certain chemistry and does not allow it to aggregate. So, here inhibition of aggregation that keeps the native structure intact. This is the native structures. And here this figure you can see this is the fiber formations of the peptide, of the beta amyloid peptide, and you use a certain dye to see that it shows green color for everywhere wherever the fiber is. Now, once you add this molecule, then the fiber is broken, because the molecule gets intercalates within inside the peptides, and then it disrupts the stacking or disrupts the aggregation of the fibers or of the peptides. There is no fiber and it is a black color, no green color. So, again, once you know the every step of it, then only you can find out about a good solution. So, we will see the structure of some of the important biomolecules now, and then later on I will discuss them in detail as we move on. So, here these are the structures of the small molecules, the vitamins that we have in our body. And left hand side you can see some structures of the vitamins, and right hand side the other kinds of structures. All the structures in the left hand side are basically the hydrocarbons, which has carbon and hydrogen mostly, and they do not have many functional groups. Of course, it has OH, but compared to the large molecule, this is a single functional group and no charge separations. So, these molecules on the left hand side are mostly hydrophobic molecules. They are hydrophobic compounds, and therefore, they are not soluble in water. In body, they are soluble in fat, in fatty acids. On the other hand, in the right hand side you can see these structures, for example, vitamin B9 is a folic acid. This has a functional group, acid group, is acid group, all heteroatoms, so many heteroatoms are there which are hydrophilic in nature. So, this overall this molecule has a lot of charge separation, it is a polar molecule and a hydrophilic. Similarly, you see biotin has also carboxylic acid end with the heteroatoms there, and vitamin B1 which is thiamine, which has lot of functional groups OH and polar groups of course. So, all these on the right hand side are polar and hydrophilic. Hydrophilic in nature. So, vitamin A, vitamin D, vitamin E and K are called non-polar fat soluble vitamins, and vitamin B9, B7, B1 and vitamin C are called the water soluble vitamins. So, folic acid and biotin are actually also they of course the vitamins, along with that they are used as tumor targeting agents also, because they have receptor tumor cells have overexpressed folic acid receptor as well as biotin receptors. So, people use folic acid and biotin as a tagging to take the drugs or to take molecules to the targeted tumor cells. Coming to little bit larger molecule, cholesterol. This is again a hydrocarbon structures which has a lot of rings, cyclic structures of course, and then open chain form. This is a fatty acid. It is a long carbon chain. It can be saturated, it can be unsaturated, and terminally it has typically a carboxylic acid, but it is a long chain. So, therefore, they are not soluble in water again. Triglycerides. Triglycerides have, it is a derivative of fatty acids, you can say. They have 3 long chains, long carbon chains, and they have a glyceride moiety. Glyceride is basically, this is glycerol, CH2OH, CH2OH. So, here there is one chain, here there is one chain, here there is another chain in this molecule. So, they are called triglycerides. And phospholipid, this molecule is phospholipid, where there is a phosphate group there, and phospholipids are usually not naturally available. They are, you can modify it by another charged species, and then it can be made soluble into water also. Another very important biomolecule is of course the protein. Now, these are protein, DNA and carbohydrates, long chain carbohydrates. These 3 are the large biomolecules or the polymeric biomolecules that we will be mostly discussing in this course. So, protein is formed out of the amino acids. This is just a rough sketch, that if you have amino acids, if you have 2 amino acids, in this case I have taken one alanine and one phenylalanine. If you have 2 amino acids, and then under certain conditions or circumstances in the biological systems, they will condense, eliminate amount of water and forms a dipeptide or peptide. This is the molecule is a dipeptide because it has 2 amino acids. So, this is the peptide bond CO and NH. This is basically an amide bond, and in this case when it is formed between amino acids, we call it a peptide bond. So, the new peptides are formed, and the sequence of the peptides, many amino acids together will conjure ultimately a 3D structure of the protein. So, they will fold because of its large structure, long structures, and they have many different functional groups, hydrophilic, hydrophobic both. So, they can interact in between each other, they can repel. So, that forms a 3D folding structures of the protein that will give you the ultimately active protein which has certain biological functions. So, this is the course content. Now, I am coming more to the real syllabus. This is how I have prepared the module. There will be around 10 modules. The first module will talk about nucleic acids and proteins. Second one is the synthesis of nucleobases and nucleosides. So, here we will see how we can synthesize the nucleobases or their derivatives that can have other properties, other biological properties as well as other pharmaceutical properties in laboratory. They are basically organic synthesis protocols, both the nucleobases and the nucleosides with the sugar, how to attach the sugar to the nucleobases. And then the third module, coming back to the biology, is DNA replication, how the process of DNA replication happens, what chemistry is involved, why it is particularly happens in that way, what is the difference between the 2 strands of DNA, what is the activity of the enzyme, key enzyme of the replication process, that is polymerase. There are other enzymes involved, we will see. And then comes DNA sequencing. Once you isolate a DNA, how can you find out what is the sequence of the DNA? And then PCR is polymerase chain reaction. PCR is a technique through which you can amplify or increase the quantity of DNA that you have isolated, and that is very important in laboratory research, because if you isolate a DNA from some source, from a disease, from other organisms, then the concentration of DNA would be very small, with which the working in the laboratory is difficult. So, you need to increase the amount, and that is the PCR is the technique for that, through which you can amplify the amount of DNA. And then fourth is the DNA damage. What are the chemical changes that happens in DNA, which happens due to when it is exposed to different kinds of internal or external chemical agents. And those damages, how are they responsible to induce mutations in the DNA, and those mutations are ultimately the cause of cancers or other genetic diseases. Then fifth module is DNA to proteins, how proteins are synthesized taking the information from the DNA or information from the genetic code, the processes of transcription, translation and the genetic code. Here a lot of chemistry is involved. We will see all the processes there, the biological process as well as the chemistry behind them. Then module 6 is peptides, how to characterize or how to find out the short peptides, that is the constituents of the proteins, how to sequence the proteins or how to sequence the peptides. You have isolated a protein sample and then of course you have to know what is the sequence of the proteins, which amino acids are engaged and what is the series of the amino acids, that is the sequence of the proteins. How to find out about the sequence of the proteins, the chemical methods as well as the biological method, and then its use in therapeutics. Module 7 will be little bit organic chemistry, that is the solution phase and solid phase synthesis of peptide, how to synthesize a peptide in laboratory. Number 8 is changing the genetic code. Now how you can develop artificial base pairs, how you can develop artificial genomes, or we call them molecular probes. That is called expansion of genetic code. There is peptide nucleic acid, as PNA, LNA, locked nucleic acid, and how they act as molecular probes in diagnosis of a disease or in curing a disease sometimes even. Module number 9 is modern techniques for biomolecules and disease diagnosis. So, nowadays we use lot of good techniques and lot of tools. There can be chemical tools, they can be biological tools, or they can be instrumental techniques to study the various aspects of biomolecules and to study the diseases also. So, I will cover some of the techniques, important techniques that are vital for chemical biologists as well as biochemists. Number 10 is the structure, chemistry of sugars and carbohydrates, and how we can synthesize the carbohydrates and what are the applications of carbohydrates in biology. So, this is roughly the course content. I will start with DNA, the nucleic acids. I think many of you can recognize this picture. Who are these people? This is James Watson and this is Francis Crick, and they are showing basically the model of the DNA. So, this is the first time that people came to know how a DNA looks like, the double helix, the structure of the DNA, and that has come out in this famous Nature paper in 1953. The title was A Structure for Deoxyribose Nucleic Acids. Now, if you look at the history or the story, background story behind the double helix structure of the DNA, then James Watson and Francis Crick, none of them actually has done any experiments to find out the structure of the DNA. James Watson was a postdoc, he came from America, and Francis Crick was still doing his PhD that time at the Cavendish laboratory, and it was their common passion and ambition to find out the structure of the DNA. Nucleic acids and its structure was a very hot topic in that time, and people were dying to know because it was known before that the DNA is involved in storing genetic information, and when cell division happens, DNA is the molecule that carries the all the properties or the information from one cell to the other and responsible for all cellular activities. So, the question was how it looks like and how it carries the genetic information from one cell to the other. When cell division happens from one cell to two new cells are formed, how a DNA is transmitting the same information in this cell as well as in this cell. So, it was very important to know the structure of the DNA, and Watson and Crick, they used to meet in a cafeteria and discuss, try to make the models of it. Of course, they are not the only people. What they have done, they have actually gathered the informations that were available that time, and those are the key information. There are many informations floating around, of course, in any science, but you have to find out, or you have to tune together the right kind of informations. So that they have done properly. So the story starts with this. Of course, they have received the Nobel Prize 1962, along with James Watson, Francis Crick, and then Maurice Wilkins. These three people. DNA was first isolated in 1869 from white blood cells (WBC) by Johannes Friedrich Miescher, and then a key development was here in 1919 when Levin has found out the composition of the DNA, that DNA consists of, it is not a single molecule, it consists of phosphate, it has sugar unit in it, and it has a nucleobase, basically a heterocyclic compound, heterocyclic base in it. So it has three key important components in it. So that was one big discovery on DNA. And then later Erwin Chargaff has found out that a very interesting fact, that if you isolate DNA from multiple resources, so by that time it was known that DNA consists of course phosphate. What is the sugar? It was established that it has a ribose sugar, and it was also established that it has 4 nucleobases, and they were termed as adenine, thymine, guanine, and cytosines, 4 different compounds, heterocyclic compounds. So Erwin Chargaff found out that if you isolate a DNA from one sample and then analyze, then the number of adenine or the number of moles of adenine that you obtain is always equal to number of moles of thymine. Similarly, number of moles of guanine or number of molecules of guanine is equivalent to that of cytosine. So that is there if you isolate DNA from sample 1, if you isolate the DNA from source 2, whatever the source is, does not matter. The DNA, all kinds of DNA has this parity in it, that number of adenine and thymine are equivalent quantity, guanine and cytosine are equivalent quantity. So that was a very important piece of information that Watson and Crick always kept in mind. And then came the proper experiment, or the most beautiful experiment done on DNA is the X-ray crystallographic diffraction, X-ray structure of a DNA. That was done by Rosalind Franklin and Maurice Wilkins in Cambridge University. So they have first found out or published the structure of the X-ray diffraction pattern of the DNA, and the diffraction pattern gave the idea that DNA has to be a symmetrical geometry. The molecule should have a symmetry in it. It is a symmetrical geometry. So that is another piece of important information, but the center of this information was this book and this man, Linus Pauling, and his famous book, The Nature of the Chemical Bond. So this is the book, it is a center in chemistry basically. So whatever the bond lengths, the single bond distance, double bond distance, the angle between the atoms in organic molecules, electronegativity scales, whatever, all those things are in these books, and this is the Bible of for chemistry. So all these informations that the organic chemistry is now stands upon is due to because of this book. And Linus Pauling has received Nobel Prize for this book and his work in 1954. So he was already a Nobel laureate, and he was in between. He is the first one who has found out about the structure of the proteins, the alpha helical structure of the protein, beta sheet structure of the proteins. So Linus Pauling was the key person behind the three-dimensional structure of proteins. So he was already a very celebrated scientist, and he was also the one who was trying to solve the DNA puzzle. What is the structure of DNA? So there are at least three important players. One is the Linus Pauling, trying to figure out the structure of DNA, and he was the most likely person because he has already found out the structure of the proteins. And then Franklin and Wilkins, and then the passionate guys, Watson and Crick. So what Watson and Crick has done, they have actually accumulated all the right informations, that is this adenine, thymine, guanine, cytosine equivalents, keeping in mind that DNA should be symmetrical. And they knew, of course, that DNA has phosphates, ribose sugar, and nucleobase. Now on that basis, they have started making the models. And when they were doing the models, they have all kept very carefully the bond length, the bond angles as for given in this book. So, using this they came up with the final structure of the DNA. It is a unique structure, or it is a unique molecule. Then only it was kind of clear that DNA is the unique molecule present in our biological body, because no other molecule is looking like the DNA, because it is not a single molecule, it has two molecules in it, and they are actually connected together by hydrogen bondings and other interactions that we will talk. So, it is basically two molecules, and that also explains how the genetic information is carried over from in two different new cells. So, this is what the structure of DNA looked like, and all the bonds and these things came from the book as given in the book. So, it shows that it has a ribose sugar 5-membered ring here that is connected with a phosphate to the 5' end. So, this is the structure. Here is a nucleobase, I am writing B for base, deoxyribose. This is what a deoxyribose sugar looks like. This is number 1, number 2, 1 prime we call, 2 prime, 3 prime, 4 prime carbon, and this is a 5 prime carbon. So, you have one hydroxyl group at the 3 prime end, another hydroxyl group at the 5 prime end, and then at 1 prime end you have the nucleobase ATGC. So, this is the 3 prime end of it with a free hydroxyl group, and then to the 5 prime this way it is connected to a phosphate, and then phosphate is connected to the 3 prime OH of the next sugar, and that is how it goes on. And of course, it has kept the angles at par the given angles in the book. This P means actually phosphate, it has a double bond O, double bond O. So, all of them. So, it is a sugar, phosphate, sugar, that is the skeleton, that makes the skeleton of the DNA, and then you have the nucleobases. And Watson Crick found out that if you attach the nucleobase to the 1 prime position, then you get a pattern that this is the axis of the DNA, the sugar, phosphate, sugar, that is the skeleton, and then the nucleobase is they are aromatic rings. So, therefore, planar systems, they are perpendicular almost perpendicular to the helical to the skeleton. So, they are like this, flat. This is the ring, this is the geometry, flat. So, when the two nucleobases are there, they are present on top of each other like this, one nucleobase, another nucleobase. So, therefore, they can come into the pi stacking interactions within the pi orbitals overlap. And similarly, on the other side, if you think of two strands here, this is the nucleobase orientation, and in this case this is the nucleobase orientation. So, they are coming in this way, two nucleobases in two strands are coming in this way. That is what it is showing here, is shown here. So, they have found out that if you do that way, then the two strands can come close enough proximity to form the hydrogen bonds. And hydrogen bonding, of course, is a key interaction that brings two molecules closer, and it is not strong enough that you cannot detach it. So, that is the beauty of DNA actually. Moreover, what they have found out is that the second strand, if you keep on the same line, same direction, so this is 3 prime direction, this is 5 prime direction. If you keep on the same direction, then the nucleobase in this strand and nucleobase in the other strand, they do not come into the hydrogen bonding distance. Once you turn it down, upside down, making it 3 prime on this one up and 5 prime down. So, one is 3 prime to 5 prime, other one is 5 back 3 prime to 5 prime back here, opposite orientations. Then with the sugars faces down, then the nucleobases are actually coming within proper hydrogen bond distance. enormous amount of chemistry and knowledge in chemical structures are present in the overall structure of DNA and functions of DNA. So, and parallel strand and antiparallel strand that govern the hydrogen bond distance, hydrogen bondings between the nucleobases in the opposite strands. And along with that, as I said, that within the same one strand, two nucleobases are on top of each other. So, they are pi orbitals can overlap, that gives more stability for DNA. It is called pi stacking. So, DNA is the genetic storage unit, RNA is the genetic storage unit in virus, and they also RNA also act as catalyst and gene regulator. So, most naturally occurring DNA is double stranded DNA, which is the structure of which was given by Watson and Crick, locked in a helical structure, and they are of course consist of deoxyribose for DNA, ribose for RNA, sugar, one unit of phosphate, and then heterocyclic nucleobases. For DNA, it is A, T, G, C. Alignment, the sugar and the phosphates along the helical axis, as I have mentioned. Nucleobases are perpendicular to the helical axis. Direction, 5' end to 3' end, other one is 3' end to 5' end. And always 3' hydroxyl is free. This is how a DNA is written. If you write a DNA, you will always see that if this is a 5', if this is a 3', 3' always ends with a free hydroxyl, 5' always starts with a free phosphate. Now, the key fact, that how does then the DNA transmits the genetic information from one cell to the new cells. Now, once you see that DNA consists of two molecules, now things become very easy. And this also explains this hydrogen bonding between the nucleobases, also explains why the chart graph has seen the number of adenine equivalent to number of thymine, number of guanine equivalent to number of cytosine, because adenine is paired up with thymine, guanine is paired up with cytosines. That is the best pairing, the strongest hydrogen bond interactions that you can expect if you do the model. So, once you know the DNA has two strands, parallel and antiparallel, and that has the ATGC base pairings, now we will call, then things becomes easier to explain how it stores the genetic informations. So, I will talk in details about the DNA replication process. Roughly just I will show, if you have the DNA strands this way with the hydrogen bonding here, then of course hydrogen bonds are not strong enough. As I have said, they are not covalent bonds. So, they are weaker bonds with slight energy. You can separate the hydrogen bonds, or you can destroy the hydrogen bonds. Once you destroy the bonds, then your DNA, two DNA strands can be separated. Now, without the direction, the DNA do not exist. Whenever you want to write a sequence or a representation of a DNA, we always have to mention the ends, 5' to 3', 3' to 5'. So, this is 5' to 3', and this is 3' to 5'. You can change the strands, and now again each strand can be acting as a template to synthesize a new strand. And of course, since ATGC pair up is inevitable, it will only pick up the complementary of it. If ATG, then this will pick up, this has to pick up T, this has to pick up A, this has to pick up C, and you will synthesize a new strand. Similarly, here a new strands will be synthesized, and that is how new strands, you have a complete DNA now, here you have a complete DNA now there. So, two new cells will have exact sequence of the DNA which was present in the parent cell. That is how the genetic information will be stored, as explained by the modeling, the double helix model by Watson and Crick. So, it is a beautiful model, one of the unique geometry of the molecule, and of course, the function is unique, that this is the only material that can replicate itself, that can make its own copies. There is no other biomolecule, no other molecule in fact, which if you keep, that will make its own copies. This is the only molecule. So, with this I will stop today, and from next class, in the next lecture, I will give some more details of the structure of DNA and then other functions, other properties. Thank you.